Hello everyone, this is Ketish here. In this video, I'm going to go through question number 3 from the AQA Biology 2022 Biology Paper 1 higher tier. This question is on the topic of cell biology, and it's actually referring to the microscopy required practical. A student prepared some onion cells. The student viewed the onion cells using a light microscope. This is the method used. Cut an onion into pieces using a sharp knife. Peel a thin layer of onion epidermis from one piece of onion. Place the onion epidermis into a microscope slide in a single flat layer. Add 3 drops of iodine solution. Slowly lower a cover slip at an angle onto the onion epidermis. Place the slide on the stage of the microscope. Table 4 shows a risk assessment for this experiment. Complete table 4. So you have the hazard, which is the iodine solution is an irritant, and the sharp knife, and the risk for the iodine solution may cause allergic reaction or skin rash. Plan to minimize risk. All you need to say is what happens if you get it on your skin. So you could say wash skin immediately after contact, or wear gloves, clean up spills, or use a dropper bottle. Sharp knife, what is the risk with a sharp knife? Well, you may cut yourself, so may cut you or someone else, or it may pierce the skin. What is the plan to minimize the risk? Make sure you handle it carefully. You could say cut away from the body, or cut on a chopping board, or keep fingers away from the blade when cutting. Next part, 3.2. Give a reason for each of the following steps in the method. A thin layer of onion epidermis is used. Why would you want to use a thin layer? So that you can see individual cells. Now, if you have a thick layer, you're not going to be able to see a clear image of the cell. So you really want to just have one layer of cells to see it clearly. Iodine solution is added to the onion epidermis. The iodine is actually used as a stain to see parts of the cell. So what you would see is something very similar to this once it's been stained with iodine. The cover slip is lowered onto the onion epidermis at an angle. Why do you want to put it at an angle? Is to prevent or reduce air bubbles. If you just lay it on the top, it might actually trap air bubbles. So here's an image of what it will actually look like. So if it had air bubbles, these are the air bubbles which has actually been trapped underneath, right? And this part is showing basically the layer actually folded together. Figure 3 shows what the student saw under the microscope at a magnification of 400. So the length of cell Z in figure 3 is 4.8 centimeters. So you got cell Z, which they have given as 4.8 centimeters. Calculate the real length of cell Z. Give your answer in micrometers. So you should remember the cell magnification equation for this. You're not going to be given this unless you are doing the foundation paper. So the easiest way to do that, because they want the answer in micrometers, I'm going to convert this length, which is 4.8 centimeters, to micrometers first of all, and then find the cell length. So 4.8 centimeter is actually 48 millimeters multiplied by 10. Now remember, 1 millimeter is equal to 1,000 micrometers. So if I convert this 48 millimeters to micrometers, I just need to multiply it by 1,000. So multiplying that by 1,000 gives me 48,000 micrometers. Now I can use this value in the equation. So what is the equation for magnification? Magnification equals this image size divided by the let's call it the size of real object. So in order to find the real length or the size of the real object, say size of real object, you would, let me bring that up a little bit higher, right? So image size divided by the magnification. So that's 48,000 divided by 400. So let me bring up a calculator. 48,000 divided by 400 gives me 120. So that's the answer is 120 micrometers. Next part, figure 4 shows the students drawing in figure 3. So they have called it the onion cells and they have drawn a simple diagram. Give 2 ways the student could improve the drawing in figure 4. Now there's no other labels in the diagram and there's a little bit of gaps. There's gaps in the lines. It shows just one line. So if I bring that, so this is what you would actually see. So if you're trying to draw this diagram, then you would want to make the lines a bit thicker to show the cells, and you can see that there's no gaps in between. They're actually very compact. So here's some suggestions to improve, right? Include the magnification of the scale. Any of these 2 points will give you the 2 marks. Use continuous lines to ensure no gaps in the lines. Do not draw overlapping cells or draw wider cell walls. Do not shade or draw all the cells present. Draw correct cell shapes or do not have gaps between cells. Draw nuclei in correct location or label the cell parts. Now, the lines that you see here is not actually the cell membrane, it's actually the cell wall that is actually being stained. So you'd want the lines to be a bit more thicker. Onion cells can be seen using an electron microscope. Now you need to know the difference between an light microscope and an electron microscope. In schools, what we use is actually just a light microscope. We don't have access to an electron microscope. They are very expensive. And you need to know what are the advantages and disadvantages of each of them. So give 2 ways onion cells would look different when seen using an electron microscope. Electron microscopes, the magnification is way much higher. So would look much more magnified or bigger. The cell would have more detail or it would have a higher resolution. Or you could say that you could see more subcellular structures in more detail. That brings us to the end of question number 3, and I will see you in question number